Let's learn about the electron transport chain. So first, let's orient ourselves within the cell. So the electron transport chain occurs within the inner mitochondrial membrane of the mitochondria within the cell. So here's our inner mitochondrial membrane, and you can see this is the electron transport chain. And then we have the mitochondrial matrix on the inside, and then the intermembrane space on the outside of that inner mitochondrial membrane. So now that we know where we're at, let's look at the exact function and mechanism of how the electron transport chain works. So as you can see here, we have these complexes, right? And the first one we have is aptly named complex 1. So what happens here is we have a molecule of NADH, and it gets oxidized to NAD+. So oxidation means that we've lost an electron, which makes sense since this product is now positively charged. But where did that electron go? Well, the electron goes onto complex 1. And the way you can think about this is when that electron goes to complex 1, it kind of supercharges it or gives it a superpower. And what that power is it gives complex 1 the ability to pump protons from the mitochondrial matrix into the intermembrane space. And specifically, complex 1 can pump 4 protons into the intermembrane space with each NADH that gets oxidized. So once these electrons do their function on complex 1, they get shuttled over to coenzyme Q. And then next we have complex 2 here. Now complex 2 does not have the ability to shuttle protons across into the intermembrane space. And when we look at the structure of these different complexes, it kind of makes sense why. So as we can see, complex 1, 3, and 4 are all shuttling protons into the intermembrane space. And it makes sense because they span the entire membrane. So if we look at all of these, they span the entire membrane. So as a result, they have the ability to take protons from the mitochondrial matrix and send them into the intermembrane space. Whereas complex 2, as you can see, does not span the entire membranes. As a result, it doesn't shuttle protons into the intermembrane space. So remember, complex 2 is the only complex that's not involved in sending protons into the intermembrane space. Now, what happens here is that FADH2 gets oxidized to FAD. It puts its electrons onto complex 2, and then those electrons get sent onto coenzyme Q. So now we have the electrons that were used up by complex 1, and then the new electrons from FADH2 that get sent to complex 2, both are now onto coenzyme Q. Next, coenzyme Q shuttles these electrons onto complex 3. And then the same thing happens again. Complex 3 now gets supercharged because it has those electrons, and it's able to pump 4 protons from the mitochondrial matrix into the intermembrane space. Now those electrons have done their job. They get shuttled over to cytochrome C, and then lastly they get sent to complex 4. Now, as we can see, complex 4 does the same thing as complex 1 and 3. It's going to send protons from the mitochondrial matrix into the intermembrane space. But the one difference that we might notice is that complex 4 can only shuttle 2 protons across, whereas 1 and 3 were able to shuttle 4 protons across. How do I remember this? Well, the electrons from NADH, they get put onto complex 1, and then some of that magic or energy that they have gets used up in order to pump those protons across. Then they get sent over to coenzyme Q. Well, then we get a new set of fresh electrons that haven't been used up from FADH2. Those go into coenzyme Q, and then together they get sent onto complex 3. So still we have those fresh electrons from FADH2, so they still have that juice, right? They still have that ability to give a complex 3 the full function of putting across 4 protons. But when we send those electrons now to cytochrome C in complex 4, we're not getting a fresh set of electrons anymore, right? So the way I like to kind of remember it is the electrons that are used by complex 4, some of that magic or some of that energy has been used up now, so it can't pump across as many protons as complex 1 and 3 can. So as a whole, we have 10 protons that get pumped across, 4 by complex 1, 4 by complex 3, and then 2 by complex 4. Now, what is the point of all of this? And actually, before we get to that, I want to take a step back real quick and mention NADH and FADH2, they come from a very specific place that we need to think about. These are products of the Krebs cycle. And I mentioned this in my Krebs cycle video as well. So if you're unaware of that, go back and review that video. But the reason I mention this is because I think it puts into perspective that these metabolic processes are not happening independently of each other, right? The products that are made as a result of these processes are made for a specific reason, right? And NADH and FADH2 were made by the Krebs cycle, so then we could use them in the electron transport chain to then do this process that we've been discussing today. Now, the whole function of the electron transport chain is to pump protons into the intermembrane space. Why have we done that? Well, a very important mechanism to understand for the MCAT and just biology and biochemistry in general is concentration gradients. So things will naturally flow from areas of high concentration to areas of low concentration. So we have set up here a concentration gradient by pumping all the protons into the intermembrane space. How did that happen? Well, I have a ton of protons now in the intermembrane space and not as many in the mitochondrial matrix because I took all of them from the mitochondrial matrix and put them into the intermembrane space. So I have a bunch up here and not as many down here. So I've created a concentration gradient. So naturally speaking, the protons are going to want to move from areas of high concentration, as in the intermembrane space, to areas of low concentration, such as the mitochondrial matrix. I can use the ability of protons here wanting to flow down their concentration gradient in order to make ATP. And the way we do that is we have another molecule called ATP synthase, right? And it has the ability to let protons flow through it. So what happens is in ATP synthase, and again, this is all happening in the same location, I just added this onto the drawing, is that ATP synthase lets protons flow from the intermembrane space into the mitochondrial matrix, right? So it's flowing back down its concentration gradient. And since it's going down its concentration gradient, this does not require any energy to happen. It'll just naturally happen this way. So I couple that, the natural flow of protons wanting to go back to the mitochondrial matrix, with the ability to turn ADP into ATP. So this is how ATP synthase works. So that way, I'm now making ATP by just naturally harnessing the energy that's created as a result of H+ wanting to naturally flow back into the mitochondrial matrix. So this is how the electron transport chain creates ATP, and also why I pumped protons out into the intermembrane space in the first place, right? The reason I did this is so that they'd want to naturally flow back, and then I could use that energy to make ATP. One last thing I want to mention here is that as these electrons are flowing through the electron transport chain, the final place that they end up is on oxygen, right? So a very high yield fact to know about the electron transport chain is that oxygen is the final electron acceptor of the electron transport chain. Then once they're onto oxygen, they'll combine with some protons, and then that'll make water. Okay, so the final electron acceptor is oxygen. So this is how the electron transport chain works. This is the way we make ATP from it, and then why we're pumping these protons into the intermembrane space in the first place. I hope that was helpful, and if you have any questions, please leave them in the comments.